Now, most of you will not still understand what a neurotransmitter is. There are until or maybe later studies, because you guys are in the sciences and you don't really pay much attention to things like this, apart from those in that are in the pharmacy and the health sciences. Then we have sodium potassium ATPase. Sodium potassium ATPase is a mode of it's a special transport across the cell membrane against the concentration gradient. That's the reason why it uses energy in form of what? ATPase, right? There are channels that help to transport sodium and glucose against their concentration gradients using what? Energy. So they call them the active transport mechanism. Active transport mechanism. But like I told you, you don't need all this knowledge. Just memorize. Is that okay? So that is that. Now we have simple proteins. Now these are proteins that on complete hydrolysis yield only amino acids. Those ones that on complete hydrolysis yield only amino acids are simple proteins like ribonuclease A and chymotrypsin. When you break them down, the only thing that they are going to yield is what? Amino acid. Those ones they are called simple proteins. And then you have albumin. Albumin is a globular protein that is found in animal and plant tissue. Ovalbumin from egg. Lactalbumin from milk. Serum albumin from blood plasma, and legumelin from plants. Now these are just the different class of albumin. We say albumin are globular proteins found in animals and plant tissue. Example is ovalbumin from egg. Lactalbumin, like lactose. You know, when we talk about lactose, we talk about milk. Then serum albumin from the serum of the blood, call them serum albumin, which is plasma serum. Then legumelin, from legumes, you have plant. Is that okay? Then derived proteins. These are proteins product from simple and conjugated proteins. Now we'll talk about conjugated proteins. What are these conjugated proteins? These are simple proteins combined with non-protein group called prosthetic group. They are simple proteins combined with non-protein group called prosthetic group. So when you have a simple protein like ribonuclease A and chymotrypsin combined with another group, you call them what? Conjugated proteins. For instance, I talked about lipoproteins. Lipoproteins. When you have the prosthetic group as what? Lipo. That means the other group that is attached to the protein is called the prosthetic group. So for instance, you have lipoprotein. The prosthetic group here is what? Lipo, which is lipid. Glycoproteins, like I told you, it's conjugated with protein. The prosthetic group here is what? Glyco. Hematoprotein. The prosthetic group here is what? Blood. Hema. Whenever I hear the word hemato, have to do with what? Blood tissues. The phosphoprotein, phosphorus or phosphate group. Metalloprotein, in particular metal, bonded with what? The protein. These are the various prosthetic group. Now, the group bonded to the protein is called the prosthetic group, and then the protein part is called the apoprotein. The protein part is called the what? Apoprotein. So any other this partner is called the what? Apoprotein. Then both of them combine together, you call them what? The entire molecule, you call it what? A holoprotein. So when you say lipoprotein is a holoprotein. Glycoprotein is a holoprotein. Hematoprotein is holoprotein. Phosphoprotein is what? But then when they ask you what is the prosthetic group in each of these proteins, then you can say the lipo group, which is lipid, glycol, carbohydrate, hemato, blood, phosphates, and then metallo. Any of the metals actually form the conjugated protein. Is that okay? So that is that. And we said proteins are built from 20 different amino acids. True or false? So where you have the amino acid, I've actually explained this before, so I wouldn't spend much time explaining this particular one because I've explained this before. So when you have an amino acid, and that amino acid, let's say you are taking any amino acid, you have something like this in your material in page 9. This is an amino acid. I've explained this before. This is another amino acid. Now, when these two amino acids combine, let's write the product under. What did I say will happen, my dear student? Told you that water molecule will be what? Given off. And where water molecule is given off, that means you take one hydrogen from here and take OH from here. You are going to form a product. The product you are going to form will be H2N CHR CONH CHRCOOH. Now, we said that this linkage that is formed here is called a peptide bond, right? This is called a what? Peptide bond. This is something like that. Exactly, we said it. Now, and I told you that each of these amino acids that are left, the part of the amino acid that is left, is called a residue. So this is residue 1. This is what? Residue 2. They are also called what? Peptide. So here you have peptide 1, and here you have what? Peptide 2. So in total, what do you call this molecule here? You call this a dipeptide. You call this a what? A dipeptide. Are you getting it now? So that is that. Now, there's a way you can classify peptide. Are you getting it? I've actually explained to you. If you have 3 of these amino acid residue, you call them tripeptide. For instance, if I now add another amino acid here, which we can also express as something like this. You can express this as something like this. It's still the same thing, all right? Don't mind. This is me bringing this group out. This CONH. This how you can actually write it. C double bond ONH. And they must be trans to each other. The oxygen and the amine hydrogen, which is this oxygen here, this one here, must be trans to each other. Is that okay? Now, this now a tripeptide. Why? Because it has 3 residue. This residue 3. This peptide 1, 2. This peptide what? 3. If I add another one, it become what? A tetrapeptide, pentapeptide, and then until you now have dozens to dozens to millions to billions to thousands to thousands to hundreds to hundreds. Those ones are called what? Polypeptide. And those are what form proteins. Do you understand the concept now? So that is that on the peptides. Like I told you, if you have plenty of them, you have called them polypeptide. Now, how do you now name them? Depending. If this thing consists of tyrosine, you say you don't call it. Let's say the peptide consists of tyrosine, aspartic acid, and let's say glycine. Let's say it starts with alanine. You know, each of these are amino acids, Abi. Let's say you have these amino acids joined together. What are they going to form? 1, 2, 3, 4. They are going to form what? What kind of peptide? 1, 2, 3, 4. Tetrapeptide, right? Exactly. So this one now, you are not going to call it alanine, tyrosine, aspartate. How do you call the peptide now? It's now going to be alanyl. This INE, change it to YL. Is that okay? So this gives us aniline. This tyrosine, INE, change it to what? IL. YL. This tyrosine. This aspartic, IC, change it to IL. So this is aspartyl. And then the last one, you write it in full, glycine. Alright? So these are like substituents, alkyl substituents, alanine, tyrosyl, aspartyl, glycine. Are you getting it now? So if histidine is the one that ends it, you will still continue. This glycine now, remove this group here. So IN turns to YL. So this glycyl, and then you have what? Histidine. So this is how to name polypeptide. Alright? Exactly. Now, another thing that you also need to know is that most times they interchangeably differentiate between they use polypeptide and protein interchangeably, but proteins and polypeptide can actually be used interchangeably because we said that proteins are polypeptide chains. All right, so you can actually use them. But proteins are broadly molecules that contain one or more polypeptide chain. It does not necessarily mean it is just one polypeptide. It can be more than one polypeptide chain. Now, proteins having only one polypeptide chain are monomeric protein. So if a protein is having just one polypeptide chain, let us say this is the chain. A protein that is having just one polypeptide chain is said to be what? Monomeric protein. This is another exam question also. You'll be asked, explain the following concept. All right, so any protein that have just one polypeptide chain is a monomeric protein. Now, proteins that are composed of more than one polypeptide chain are multimeric. So if you have something like this is just one polypeptide chain. That means it consists of different amino acids joined together, right? Monomeric. Now, how about if it has more than one, like insulin structure has two polypeptide chain of about 50, 51 amino acids joined together by disulfide bond. Now, this is multimeric protein because it has two polypeptide chains. It could have more. It's not just two. But whenever the polypeptide chains are more than one, you call it a multimeric proteins. Now, multimeric proteins may contain only one kind of polypeptide, in which case they are homomultimeric. Now, what do we mean by that statement? You said if a polypeptide has only one type of polypeptide chain is monomeric, right? If it has two types of polypeptide chain is what? Multimeric. Now listen, if the two polypeptide chain are the same, for instance, let us say the two polypeptide chain are this, they are exactly the same polypeptide chain. Let's say what is in chain A here is still the same chain that is here. So two the same chain. Are you getting it? But they are two chains joined together to form the protein. You call them, of course, they are multimeric because there are two chains. Homo because they are the same. So you call them homomultimeric. You will be asked to explain this. Homomultimeric, explain the following term: homomultimeric, heteromultimeric. So if you have two chains A and A, they are the same, but they are two chains. Two chains make them to be what? Multimeric. The same make them to be what? Homo. Now, however, you could have a situation where you have, this is chain 1. What do you call this? Monomeric. Mono means what? Mono means 1. Now, this could be chain 2. And obviously, this is B. This is what? A. Chain A is different from chain B. Of course, they are what? Joined together by some kind of bond. Now listen, chain A and B are different. So, because they are different, first of all, different, you use hetero. There are two chains. Two chains make them to be multimeric first, but because they are different, you now call them heteromultimeric. Heteromultimeric. Do you understand? So that is that. Now, let us look at some biological active or biological important peptide. I really wish to explain this in detail, but time is against me. Plus the fact that you can go and read about this. This is a drug. It's called glutathione. Now, this glutathione is a tripeptide of glutamate, cysteine, and glycine. It is a tripeptide, so it is not a protein per se. It is a tripeptide. When we talk about tripeptide, it means it has 3 amino acids in it. The 3 amino acids are glutamate, cysteine, and glycine. So if you follow this order, what are you now going to name them? Are you getting it? You can say it's glutamyl, cysteine, glycine. All right. Now, this glutathione, its function is to destroy harmful oxidizing agents in the body. Hence, they are called antioxidants. Now, what are these antioxidants? And what are these oxidizing agents? Now, your body, in response to stress, some environmental